Okay, so what I want to go over today is the cerebellum. So looking over the anatomy, looking over the physiology, and then at the end we'll go over DANISH, which is a mnemonic for the clinical signs that somebody might present with if they have a cerebellar lesion. So we'll just get straight into it. Cerebellum means little brain. It's this bit over here, and it contains over 50% of the neurons. And the interesting thing about the cerebellum is that it doesn't create, people think of it as associated with movement, but it's not the thing that creates movement. So here, just draw that line as the central sulcus, um, there, and then over around here, in front of that is where you're going to get kind of the ideas for creating motor plans. What the cerebellum does, it just coordinates and fine tunes those movements. So by having a cerebellum, rather than something being a very jaggedy movement, it just makes it a very smooth, flowing action. Okay, so here we've got the cerebrum, here we've got the cerebellum, and this bit here is the brainstem. Here is the midbrain, pons, medulla, and then the thing to know, so the reason why the pons is highlighted is that because the cerebellum is at the level of the pons, there is like a sheet of dura mater here called the tentorium cerebelli. It just divides the occipital lobe and the cerebellum. These circles here are called the olives. So if I just draw a little quick diagram of the brainstem, so kind of looking at it from that front angle, as though if you're looking straight at someone and you're looking straight through at the front of their brainstem. So if that's the pons, here's the pyramids. This is now in the brainstem, in the medulla, sorry. There's your midbrain, and then you've got these things kind of here, which are the olives. So here, midbrain, pons, and then the medulla oblongata. This bit here, there's the pyramids, you can get corticospinal tracts there, but here, these things at the side are called the olives. What the olives do is they contain these things called the olivary nuclei, so superior and inferior olivary nuclei. When revising cerebellum, you'll be focusing on the inferior olivary nuclei. We'll come back to that later though. So, okay, so now going on, again, just orientation. That bit there is also the fourth ventricle. So that's in relation to, for example, with open and closed medulla. Going on to the next bit. So what we've done now is just zoom in on the cerebellum, and we've just done a sagittal cut to have a look here. What we can see here, just to orientate your pons, and then this bit here will be the medulla, above would be the midbrain, and then you've got the cerebellum. You can see kind of this branching pattern. This is called the tree of life, the arbor vitae. And this is now looking at the cerebellum from an anatomical perspective, so anatomical divisions. What you have here is the anterior lobe, here is the posterior, and here is floccular nodular. So if I just highlight that, here is the anterior lobe, this is the posterior lobe, and then this here is the floccular nodular lobe. Okay, so this is now just looking at purely in terms of just how you're, from a visual perspective, how you're just dividing up the cerebellum. Also something to note, here you've got the cerebellar tonsils. The thing about cerebellar tonsils is that the cerebellum sits at the bottom of the brain sits inside the skull, and inside the skull, the brainstem is going to pass through, it needs to pass through to get to the spinal cord, right? So to pass through, it needs to go past through a big hole. The name of the Latin for a big hole is just foramen magnum. And so what can happen is, if at this level is the foramen magnum, what can happen is the cerebellar tonsils can herniate through the foramen magnum, and they can compress here on the medulla. Medulla controls breathing, so if someone's cerebellar tonsils herniate, it can affect their breathing. So now let's move on. So all of this is just kind of more of a anatomical division of the cerebellum. Oh, and then also you've got the different fissures. So primary fissure here, just dividing the anterior and posterior lobes. And then here you've got the posterior lateral fissure, which divides floccular nodular lobe and the posterior lobe. So if you think of your cerebellum as your fist, what we're going to do is just kind of, if you just uncurl your fist now, and then looking at it as though you're looking at the back of someone's head into their cerebellum, but you just uncurled it. And what you get is something like this. So here you've got, this is more of a functional division of the cerebellum now. And what you get is kind of three different zones. So you've got here, the vermis, which is kind of in the middle. Then to the side of it here and here is the intermediate zone. So intermediate because it's between the vermis and the other bit. And the other bit is called here, which is the lateral hemispheres. So on both sides. Now you're also going to come across different, and then here you've got the floccular nodular lobe. You also come across these other words, such as like vestibular cerebellum, spinocerebellum, and pontocerebellum. Pontocerebellum, you might also come across the name of cerebrocerebellum, but pontocerebellum is more accurate. So what that kind of looks like, is, so if we say this bit here is the spinocerebellum, and then we go kind of the lateral hemispheres is then the pontocerebellum. Then floccular nodular is then going to be the last one, which is vestibular cerebellum. Okay, so what all of these basically mean is just in terms of, it's just trying to divide up what the actual function of those different parts of the cerebellum do. So if we start off with vestibular cerebellum, vestibular means balance, cerebellum means cerebellum. So this is all kind of in terms of balancing. The way that this is going to work is it will come through, you have the vestibular labyrinth in your ear, that then travels to the vestibular nuclei, and then that can go to the floccular nodular lobe. The way I remember this is because it kind of goes along in this kind of direction, and it's related to balance, and it's also related to like eye movements as well. I think of it as kind of, if it's related to balance, this is like the pole that a tightrope walker uses in order to keep balance. So then you got the spinocerebellum. It's kind of here. What this is going to do, I think this, because spinocerebellum, spine in the middle, where your spine is, and this is kind of like sensory information from your limbs. And what it does is kind of also like proprioception. So proprioception is kind of, it's described as your sixth sense. So if you close your eyes, you know exactly where your arm is in space. You don't need to be looking at your arm to go, oh, there it is. It also does like error correction as well. And then you got the last one, which is pontocerebellum. So that's kind of these bits here. Pontocerebellum is more like finer movements. And so kind of actually like when you're moving your hands in a very clear, distinct way, that's kind of coming from this bit. Where I remember that is ponto, so like pointing. So you point over there to a precise location. So point to a precise location is pontocerebellum. And that's going to be, if you point to the side, that's going to be these sides here. Okay, so then going on from here, the next thing to talk about is probably an overview of what the cerebellum looks like in practice. So if we kind of just draw out a quick diagram of the brain, and then you've got the brainstem, so it's not going to be to accurate size. Okay, what I've just drawn here, these are called cerebellar peduncles. Peduncles think of it as a bridge between the brainstem and the cerebellum. There are three, so there's superior, middle, and inferior, but I'm just going to draw two for simplicity's sake. And then there's your cerebellum. Okay, so cerebrum, brainstem, cerebellum, and then peduncles, just as a connecting bridge between the brainstem and cerebellum. So if I draw a line here, that is your central sulcus. Going to think of an idea. Say you want to move your hand towards a water bottle, and you want to pick up a water bottle. Create that idea. It then goes down. So if you're using, for example, your fingers, that kind of fine control of your fingers might be going down, for example, the corticospinal tract, in order to know then how much pressure and how much force your fingers need to when actually touching the water bottle, how much force to hold the water bottle so it doesn't slip through your hand, but you also don't crush it. So that then travels down, and then that goes to, it's going to be a really bad drawing, the hand there, and then you've got your water bottle there. Okay, where the cerebellum comes down now, and that goes corticospinal down there. Where the cerebellum comes in is that as you're moving your hand closer and closer to that water bottle until eventually you're holding it, as you're moving it this way, there is constant feedback to the cerebellum. The cerebellum is telling you how close you're getting, how much more distance it is. It's fine-tuning that movement so that you either don't just grab air because you've gone too close, you've tried to grab the water bottle too early, or you've just shot straight past the water bottle, or you just don't crush it, or it slips out of your hand. It's coordinating, it's giving you that feedback. So the way that works is it kind of comes down off these tracks going down here, sends off collaterals into the cerebellum, which then goes to, I'm going to call this DCN, deep cerebellar nuclei. These tracks can come in, and then they will go to the cortex, which is kind of this bit here, and then it can also send off these kind of collaterals again to deep cerebellar nuclei. What happens then? That information is kind of processed, the cerebellum does its magic, and then it leaves, rejoins those tracks, giving that information again. And that kind of loop is how the cerebellum works. You'll see here, stuff happens in the DCN, and stuff happens at the cortex. So hopefully that kind of explains the role that the cerebellum provides. This bit here is oversimplified. Obviously, you're going to have things like structures like the basal ganglia, which are promoting movement or not promoting movement, and structures like the thalamus, the thalamus is kind of like a toll station for the brain. Information goes to the thalamus and it sends it off to the correct positions, basically. So what we're going to do now is focus in on the deep cerebellar nuclei, what they are, and then we'll go into the cortex. Okay, so if I draw this kind of structure, it's not great, and let's go there, here's the midline. Let's imagine it's symmetrical. The way I remember the deep cerebellar nuclei, this is just looking into the cerebellum, so kind of around this kind of level, around there, because it's deep. What you have is, these are the nuclei. Okay, a good way to remember it is, don't eat greasy food. So each of these stand for the nuclei. Here is dentate. So the dentate nucleus. This one is emboliform. This one here is also symmetrical. This one is globose. And the last one here is vestigial. Okay, so these are the different cerebellar nuclei: dentate, emboliform, globose, and vestigial. This one here is on the lateral side, so this is related to pontocerebellum. These two, emboliform and globose, kind of go together. You might hear the name interposed nuclei. Those go together for spinocerebellum. And then vestigial, which is kind of in the middle, goes to here. And the way I think of it, they kind of go together like that, which are like eyes. And so then that helps me remind me that, vestigial and the vestibular cerebellum is related to extraocular movements of eyes. So those are basically the cerebellar nuclei, deep cerebellar nuclei. And that's basically what you need to remember. They just divide up kind of like that. So this area, and this area, this middle area, this one again, and then out. And it aligns up with those functional zones. So information goes from the cortex to each of those depending on what the actual movement is. Oh, another thing to remember is that there is like in the cerebrum, there's a homunculus. There's also a homunculus for the cerebellum, and looks kind of like that, just upside down. And what it basically shows is that the middle bits here of the body happens controlled by the vermis, and then intermediate zones, these limb movements in terms of like proprioception and kind of like error correction and like sensory information, that kind of happens in these zones here. Okay, so now we will focus in on the cortex, what actually happens at the cortex. It can be a bit confusing, but hopefully shouldn't be too confusing. Okay, so there are 3 different layers of the cortex, and I'll just draw them up. So there's the top layer, there's another one, there's another one. Okay, so if we label them, it's this top layer. So just to coordinate you, this is the furthest out, this is like furthest out, like, which is like here, and then this bit, which is slightly deeper, is going to be here. So if we now just label them, the top layer is called the molecular layer. The middle layer is the Purkinje layer. And the bottom layer is the granule layer. And then I'm just going to draw deep cerebellar nuclei, which is there. Okay. So in terms of going through what the cells are, because again, cells, building blocks of everything, there are obviously going to be cells in each of these layers. What they kind of look like is, here in the granule layer, it's a granule cell. So that kind of makes sense and easy to remember. That goes up like this, all the way up to the molecular layer, and then it splits into like a T kind of shape. So one side goes this way, and one goes that way. And the thing is, so it splits into that T shape. Obviously, you're going to have more than one granule cell. And what happens is loads and loads of these branches occur. So you get these kind of cells going in this kind of direction. What these look like, this then is called parallel fibers. So, because they go in a parallel shape. And there's going to be loads and loads and loads of them. So if you, for example, like cut through like that and then looking at them in that way, they're all kind of really closely bundled up together. Okay, so the next thing to know is the Purkinje cell, which is going to be in the Purkinje layer. So that could be here, to a triangle, this shape, and then that's going to go up and branch a lot, basically. And you can see that it's touching all of these parallel fibers and having a lot of connections. So the other 2 kind of cells to remember, they're not that important, so it's okay if you forget. What they are the stellate and basket cells. So they look like here, stellate, and here, the basket cell. And what they do is just inhibitory for the Purkinje cell. So the Purkinje cell is GABA, so it's inhibitory, so whatever's going down, it's just going to leave. I thought so. Go like that. Okay. Yeah. And so these 2 here, basket and stellate, just inhibitory. Then you've got here granule cell, which splits into parallel fibers, and then here you've got the Purkinje cell, which goes down. Purkinje cell is the only cell that's going to leave the cerebellar cortex and go to the deep cerebellar nuclei. And so that's basically what is setting the scene for what happens at the cortex. So now let's actually have a look at what is going on. So you've got 2 types of fibers, mossy fibers and climbing fibers. Mossy fibers go up kind of like this. and then they'll go to granule cell. They'll also send off a little collateral to the deep cerebellum nuclei. That's not too important. From the mossy fibre going up here, goes to the granule cell, goes down these parallel fibres to the Purkinje cell, down to the DCN, out the cerebellum. And that's basically what we need to know for mossy fibres. The next one is climbing fibres. So the way that they work is it just goes straight up to the Purkinje cell, wrap around, and then the Purkinje cell kind of does its thing. Climbing fibres. And then back out to the DCN. So hopefully that then makes sense for the climbing fibres versus mossy fibres. The thing with the climbing fibres is that they're slightly different because they all come from, if you remember back from here, these are called the olives. They all come from the inferior olivary nuclei. So climbing fibre, the way I remember it is climbing an olive tree, which is in a nice way. So inferior olivary, and then it enters to the inferior cerebellar peduncle, and the peduncles are just kind of these bridges. Then the next thing to go over is DANISH. So DANISH is just a mnemonic for different cerebellar signs. And this is just some of the clinical signs that you'll see. The best way to learn this is just simply going through like YouTube and finding videos to see what it actually looks like. So the first one is dysdidachokinesia. I have no idea if I've spelled that right. Probably not. Next one, so what dysdidachokinesia is if you're just flipping your hand over, if you put your hand on top of your other hand and just flipping over, rapid alternating movements. And then someone with a cerebellar sign, they can't do that. Thing with the cerebellum that you need to remember, so with the cerebrum, there's a contralateral control. If there's a problem in the right side of the brain, it will show in the left side of the body, and vice versa. With the cerebellum, there's a decussation. Decussation just means crossing. There's a crossing twice in terms of the different tracks. So the cerebellum has ipsilateral control. So if there's a problem in the right side of the cerebellum, the right side of the body is what's going to show symptoms. So the next one, so dysdidachokinesia is D, and A is ataxia. And then with ataxia, that's basically just gait, so walking in a different way. And there are different types of gaits, for example, like scissor gait. The next one is nystagmus. What nystagmus is just like your eyes kind of flickering rapidly. Thing to remember with nystagmus is just because somebody has nystagmus, it doesn't mean that they have a cerebellar lesion. There are other causes of nystagmus. If they just have nystagmus, though, it's something to investigate. So it's just your eyes kind of flicking this way and that way, and this way and that way. Next one is intention tremor. What this is, for example, let's say you want to point to your water bottle from earlier. As your finger is getting closer and closer to the target, your hand starts to have more and more of a tremor. It starts to shake more. Again, watching a YouTube video is the best way to kind of see it. S is slurred speech. I think that's fairly explanatory of what kind of happens. Your speech slurs. You're not speaking in a clear way. And then the last one is hypertonia. Hypertonia, hypo meaning low, tonia meaning like muscle tone is low. So someone might look a bit like a rag doll, their limbs are a bit floppy. So yeah, if there is one thing that you take away from this video, let it be DANISH. If you're stuck in an exam and you can't remember anything, but you just see something about the cerebellum, just list off some of these and hopefully you'll get some marks. But yeah, Hopefully that explains and gives a bit of an overview of what the cerebellum does.